Hello everyone, my name is Kushal Ingde and today we will discuss about the mitosis. First of all, we should know the meaning of the mitosis. So, mitos means the thread and osis means the state. It means that the mitosis, it is the state in which we can observe the chromosomes in thread-like form. First of all, mitosis is a specific process of nuclear division that confirms each of two daughter cells receives a diploid complement of chromosomes identical with the diploid complement of the parent cell. Mitosis is usually accompanied by cytokinesis, the process in which the cell itself divides to produce two daughter cells. The steps of mitosis are in the same in all organisms and the basic process is remarkably uniform. First is that each chromosome is already present as a duplicated structure at the beginning of nuclear division. Second, each chromosome divides longitudinally into identical halves that become separated from each other. And third, the separated chromosome halves move in opposite directions and each become included in one of the two daughter nuclei that are formed. So, this is nothing but it is the basic theme behind the mitosis. In a cell not undergoing mitosis, the chromosomes are not visible with light microscope. The stage of cell cycle is called interphase. In preparation for mitosis, the genetic material that is DNA in the chromosomes is replicated during a period of interphase called as the S phase. This S, nothing but it stands for the synthesis of DNA, during which replication process occurs. DNA replication is accompanied by chromosome duplication. Before and after S, there are periods called as G1 and G2 respectively, in which DNA replication does not take place. The cell cycle or life cycle of a cell is commonly described in terms of these three interphase periods followed by mitosis, that is M phase. The order of events is therefore G1, S, G2 and M as shown in this figure. In this representation, cytokinesis, that is the division of cytoplasm into two approximately equal parts containing daughter nuclei, which is included in the M period or mitosis period. The length of the time required for complete life cycle varies with cell type. In higher eukaryotes, the majority of cells require from 18 to 24 hours. The relative duration of different periods in the cycle also varies significantly with cell type. So it depends on the cell that how much time it takes. Mitosis requiring from half hour to 2 hours is usually the shortest period, which is found generally in embryo. Now, the mitosis is conventionally divided into 4 stages, that is prophase, metaphase, anaphase and telophase. So first of all, we will learn about the prophase. Prophase. The beginning of prophase is marked by condensation of chromosomes to form visibly distinct thin threads within the nucleus. Each chromosome is already longitudinally double, consisting of two closely associated subunits called chromatids. The longitudinally divided nature of each chromosome is readily seen later in prophase. The chromatids in a pair are held together at a specific region of the chromosome called as the centromere. As prophase progresses, the chromosomes become shorter and thicker as a result of complex coiling. At the end of prophase, the nucleoli disappear and the nuclear envelope, a membrane which surrounds the nucleus, also disintegrates, means it will disappear. Then metaphase. At the beginning of metaphase, the mitotic spindle forms. The spindle is a bipolar structure consisting of fiber-like bundles of microtubules that extend through the cell between the poles of the spindle. Each chromosome becomes attached to several spindle fibers in the region of the centromere. The structure associated with the centromere to which the spindle fibers attach is technically known as kinetochore. After the chromosomes are attached to spindle fibers, they move toward the center of the cell until all the kinetochores lie on an imaginary plane equidistant from the spindle poles. This imaginary plane is called metaphase plate. Aligned on the metaphase plate, the chromosomes reach their maximum contraction and are easiest to count and examine for differences in morphology. Proper chromosome alignment is an important cell cycle control checkpoint at metaphase in both mitosis and meiosis. In a cell in which a chromosome is attached to only one pole of the spindle, the completion of metaphase is delayed or becomes stopped. Next, there is anaphase. In anaphase, the centromeres divide longitudinally and the two sister chromatids of each chromosome move toward opposite poles of the spindle. Once the centromeres divide, each sister chromatid is regarded as a separate chromosome in its own right. Chromosome movement results in part from progressive shortening of the spindle fibers attached to the centromeres, which pulls the chromosomes in opposite directions toward the poles. At the completion of anaphase, the chromosomes lie in two groups near opposite poles of the spindle. Each group contains the same number of chromosomes that was present in the original interphase nucleus. Then moving to the next, that is telophase. In telophase, a nuclear envelope forms around each compact group of chromosomes. Nucleoli are formed and the spindle disappears. The chromosomes undergo a reversal of condensation until they are no longer visible at discrete bodies. The two daughter nuclei slowly assume a typical interphase appearance as the cytoplasm of cell divides into two by means of a gradually deepening furrow around the periphery. Now moving to the next and last, that is cytokinesis. So during this, mitosis ends with division of cytoplasm. Obviously, cytokinesis means cyto means the cytoplasm and kinesis means the division. Till now we learn karyokinesis. Now about the cytokinesis. It starts toward the middle of the anaphase and is completed with the telophase. This cytokinesis differs in plants and animal cells. In plant cells, it usually occurs by cell plate method, whereas in animals it takes place by the cleavage. Now, the significance of mitosis. So first one is that growth and development. This mitosis is very useful for the animals because a full-grown organism is developed from a single-celled zygote by repeated mitotic divisions. Next one, that is genetic stability. All the daughter cells of a multicellular organism have the same number and type of chromosomes as parent cells due to equitable distribution of the chromosomes. Repairing and healing. The mechanism for replacing old and worn out cells and healing of a wound by new cell production depend on mitosis. And next and the last, regeneration. Some organisms are able to regenerate their missing parts of body or whole organism through mitosis. So it was all about the mitosis. So I hope that you have enjoyed this video. Thank you so much.